All right, hey everybody, we're going to start our really super fast unit on plants and animals. We're going to start talking about plants, specifically angiosperms, which are the flowering plants. And we're going to do real quick an overview. So angiosperms are flowering plants, so they need to attract pollinators. They do this using visual cues or chemicals. They are able to reproduce sexually and asexually. They commonly form symbiotic relationships with other organisms. And we've been working with angiosperms for a long time. Plant breeders genetically manipulate them through artificial selection, and now we have sort of even more sophisticated ways of doing that to make GMOs. So the angiosperm life cycle we need to take a look at really quickly. So you start out with a diploid sporophyte that produces spores through meiosis. These are going to grow into haploid gametophytes, and then the gametophytes produce haploid gametes by mitosis. Fertilization of gametes then produces a sporophyte. And so it's the sporophyte is the dominant generation. It's the actual large plant that we see. And gametophytes are reduced in size and they depend on the sporophyte for nutrients. So something to keep in mind with the angiosperm life cycle are the 3 F's. So you have flowers, double fertilization, and then fruits. So if we look at this diagram here, the mature sporophyte is this guy here, okay? And the mature sporophyte is going to produce the male gametophyte, which is the pollen grain, and then the female gametophyte, which is the embryo sac. So within the embryo sac, you have the egg. Within the pollen grain, you have the sperm. So with pollination, you're going to get the sperm to the egg. Fertilization will happen. So now you've got a zygote, that is 2N. The zygote will develop into a seed within the simple fruit. The seed will then germinate and a plant will start to grow. And now you're back to your mature sporophyte. So the sort of pink color is the 2N diploid, and then the bluish green color, that is going to be your haploid with N number of chromosomes. So flower structure and function. So again, these are flowering plants. So flowers are the reproductive shoots of the angiosperm sporophyte. They attach to the part of the stem called the receptacle. And then we will talk about floral organs. And there are 4 floral organs, and those are just the parts of the flowers. So you've got sepals, petals, stamens, and carpels. And that's starting from the outside and kind of working our way in. So a stamen consists of a filament that's topped by an anther. This is where the pollen sacs produce pollen. Again, this is the male part of the flower. Then you've got the carpel that has a long style with a stigma on top of it. That's where the pollen lands. And at the base of the style, you have the ovary that contains 1 or more ovules. And if you are looking at a carpel or a group of fused carpels, you can call that a pistil. So here is a diagram. So you've got the receptacle, which is what the flower sits on. The sepals are these modified leaves on the outside of the petals. Then you have your petals, and then inside of that you have the stamen with the filament and the anther. And then finally inside of that, you're looking at the pistil or the carpel. So down here we have the ovary, we have the style, and then we have the stigma, which is going to be what catches the pollen. Now we have several different types of flowers that we can talk about. A complete flower would be any flower that contains all 4 of the floral organs. An incomplete flower would lack 1 or more of the floral organs. So you can have male flowers and female flowers. So a male flower would only have the male parts and not the female. And then the female flower would only have the female parts. When you're looking at clusters of flowers together, it's called an inflorescence. So actually a lot of the flowers that you see, so things like daisies are actually inflorescence. When you see like the one, like it looks like one daisy flower, that's actually lots of flowers, and it's an inflorescence. So we're going to first look at the male gametophyte. So pollen develops from microspores within the microsporangia or pollen sacs of anthers. If pollination succeeds, then a pollen grain produces a pollen tube that grows down to the ovary, and then it's going to discharge the sperm near the embryo sac. And then when you're looking at this, keep in mind that that pollen grain is actually 2 cells, so it's the male gametophyte and it's the spore wall. What we're looking at here is our flower, okay, and here on this side we're looking at the male gametophyte. So we have the pollen sac, we have our microsporocytes, That's going to form 4 microspores. So there's one individual one, and then we have our male gametophyte. All right, and on this side we've got the female side developing, which within an ovule you have the megaspores are produced by meiosis. They develop into embryo sacs, which is going to be the female gametophyte. Pollination happens anytime you have transfer of pollen from an anther to a stigma, and pollination can happen by, any like of the insects or birds that we've kind of talked about previously, or it can happen by wind or water. So pollination occurs in lots of different ways, and flowers are designed to have their pollination done pretty specifically one way or another. So you can have pollination by wind, by water, by bees, moths, butterflies, flies, birds, bats. Water's on there again. But pretty much anything that's going to land on a flower is going to be capable of pollinating it. But again, the flowers are usually going to be designed to be pollinated one way or the other. So here we're looking at abiotic pollination. So this is going to be pollination through wind. Pollination by bees, so it tends to be when you see like really bright, especially yellow flowers. So this is what we see when we look at a dandelion, but this is a dandelion under ultraviolet light, and it's meant to attract bee pollinators. Here we have pollination by moths and butterflies, so you can see the flower looks very different. It's set up for a very different type of pollinator. Here we have pollination by flies, pollination by birds, so a hummingbird. The hummingbird is getting the nectar from this flower, so you can see its beak is designed to go all the way kind of through the petal to get to the nectar. Pollination then also by bats. And so flowers are designed usually pretty specifically for their pollinators. And that is, they usually evolve, that the flower and a specific pollinator will sort of evolve together so that the parts all kind of work and that the pollen is situated so that the pollinator can pick it up and drop it off where it needs to be dropped off. So how does an embryo develop? The first mitotic division of the zygote is transverse, and so what that means, it's going to split the fertilized egg into a basal cell and a terminal cell. So you can see that here you've got the basal cell on the bottom and then the terminal cell on top. And then it's going to continue to develop, so it's sort of got that longitudinal development there. The embryo and its food supply are going to be enclosed by a hard protective seed coat, and then the seed is going to enter a state of dormancy. Dormancy is really important for plants. We'll get to that in a second. In some eudicots, the embryo consists of the embryonic axis attached to 2 thick cotyledons. Then below the cotyledons, the embryonic axis is called the hypocotyl and terminates in the radicle. The radicle is essentially the root of the embryo. And then above the cotyledons, you have what's called the epicotyl. Okay, so if we look at a diagram of this, we're kind of focusing up here. So we have the 2 cotyledons. The radicle is going to be the embryonic root. Then the hypocotyl, the epicotyl. Okay, so this is like a common garden bean that has really thick cotyledons. You can also have a eudicot that has thin cotyledons, and so you can see it looks a little bit different. But again, you still have the seed coat on the outside. You still have the cotyledons, the epicotyl, the hypocotyl, and then the radicle. A monocot embryo has only one cotyledon. An example would be grass, and these do look fairly different. All right, so here you have maize, which is a monocot, and so the parts are going to be a little bit different, and we'll kind of look at that as they start to grow as well in a second here. All right, so you're going to have 2 sheaths that enclose the embryo of a grass seed. So you've got the coleoptile, which is covering the young shoot, and the colereza, which is covering the young root. Seed dormancy, which I already kind of mentioned, increases the chances that germination will occur at an advantageous time and place. And so plants can't get up and move if things are not working out well for them. And so seed dormancy just sort of ups the chances that germination will happen, usually because some environmental condition has been met. So germination will start assuming that conditions are good for the seed to kind of germinate and grow into a plant. So the breaking of seed dormancy often requires some environmental cue. It could be a temperature or lighting change, it could be a certain amount of water, it could be a certain chemical, but it's going to be some cue that signals for germination to start. So germination depends on imbibition, which is the uptake of water due to the low water potential of the dry seed. Once that happens, then the radicle or the embryonic root is going to emerge first, and then the shoot will break through the soil surface. And so this looks different when you're looking at eudicots versus monodicots. Let's see. So here you have your eudicot, so this is the garden bean. The radicle or the embryonic root is going to start to grow first. Over time, then, you are going to actually start to see some of the shoot growth or the stem growth. So here it's the hypocotyl. It's kind of forming a hook and starting to push up. That's going to keep happening. The hypocotyl will get higher. You're going to start to get the cotyledons above the soil surface, and it's going to continue to grow. It's a little bit different with the maize. But again, you're going to have the radicals start to grow first. The big difference is that you don't get like that bend in the hypocotyl as this is starting to grow. Instead, you've got the coleoptile, which is going to start to grow and break through the soil, and then the foliage leaves will start to grow. So your lecture question for today is, why is seed dormancy so important for plants? I talked about it a little bit in the lecture, so I want you guys to kind of in your own words explain to me why seed dormancy is so important for plants. Thanks, guys.